So let's draw out this process. I'm going to start us off with a cell that's going to have 2 chromosomes in it. One is going to be red and the other is going to be blue. This is a diploid 2N cell, and this is our starting point. Additionally, we can say that this chromosome came from mom and this chromosome came from dad. And it's important that we remember that each of these are chromosomes. They don't have to be X's to be chromosomes. When you have X's, that's when they are sister chromatids. Right now, we have 2 chromosomes. This is a very simple animal. But before anything else, during interphase, specifically in the S phase, we are going to double our chromosome number. So through DNA replication, mom's chromosome got replicated, dad's chromosome also got replicated. So now we're ready to start meiosis, specifically Meiosis I. So the order is going to be the same, where we have prophase first, then metaphase, then anaphase, and then telophase. So with prophase 1, I'm going to draw out the cell. I'm going to write that we have our duplicated mom chromosome and our duplicated dad chromosome. These chromosomes are homologous. What that means is that they code for the same things. That could be eye color, height, whatever is on that chromosome. So maybe right here, mom has the gene for brown eyes, and maybe dad doesn't have brown eyes, dad has blue eyes, but his gene for eye color is going to be at the same location. And because those genes are at the same location, we can call these homologous chromosomes. Now, in prophase 1, we have 4N, we have 4 chromosomes. Our normal diploid number is just having 2. So we gotta get this back to our normal diploid state. After prophase, just like in mitosis, comes metaphase. And because this is Meiosis I, we're gonna call this Metaphase One. In Metaphase 1, homologous chromosomes are going to line up together. So in Metaphase 1, our homologous chromosomes are going to be side by side. I'm drawing them huge to make it easier for us to see, and there's only 2 of them. I'm going to have my centrosomes on either side of the cell with spindle fibers connecting to the chromosomes. I just have us working with one set of homologous pairs. And when they're together like this, we call them a tetrad. You can see here that they're so close together that the homologous chromosomes start to overlap with one another. And that's okay, because this section of dad and this section of mom, they have the same genes on them. Maybe this is the section where mom is coded for one eye color and dad is coded for another eye color, but both of these contain the gene for eye color. So it's not going to be that bad for them to break and reattach. And that's exactly what happens. So as these spindle fibers shorten and pull the tetrad apart, pulls the homologous chromosomes apart, before that happens, they'll trade. And they'll trade areas that are the same. And we're going to draw this out as Anaphase One. So this centrosome is pulling on that duplicated mom chromosome, but before she goes, she takes pieces of dad with her. So maybe we have one that's, doing its own thing, and the other one has taken pieces of dad, making a Spider-Man set of sister chromatids. Something you can put, don't put this on your SOL. The other centrosome pulls on dad's duplicated chromosome, but remember, he switched some parts with mom. So one sister chromatid is going to be all blue, it's this one, and the other one is going to be mostly blue in this middle section, but when they crossed over, he stole some of the red. I'm going to write in as a note here that crossing over begins in prophase 1 and promotes genetic variation. And we will see why as we continue. After anaphase, sets of sister chromatids are going to be pulled apart from each other and they're going to go to their own sides of the cell. Remember that we call that telophase, and in this case we're going to call it telophase 1. Ooh, one of them got kind of shoved up against the side of the iPad, but that's okay. I'm going to draw in mom's chromosome first. And remember, she's taken part of dad's. And then over here, I'm going to draw in dad's. In this drawing, I have shown that the tetrad, or those homologous chromosomes, are being pulled apart, and this has occurred during telophase 1. So here's a look at Meiosis I. And then after cytokinesis, we're going to have two separate cells.